Hey everyone. In earlier videos, we've seen that the immune system is associated with longevity, but how? To dive deeper into that, today's guest is Dr. George Murphy, who has been studying immune cell changes during aging and more importantly, in centenarians. Do centenarians have a unique immune system that allows them to live longer than everyone else? If so, what are the underlying details? So with that, welcome George, and let's get into the presentation.
So hello to all of your listeners. It's like I said, it's always a great day when you can talk science, and I think this is sort of a great forum to do that. My name is George Murphy. I'm one of the co-founders of the Boston University and Boston Medical Center's Center for Regenerative Medicine, which we affectionately refer to as the CREM. So here at the CREM, we're about 70 or so like-minded researchers who are interested in all things related to stem cell biology and regenerative medicine. We're lucky enough to actually work in what we think is one of the most state-of-the-art stem cell labs in the world. This is sort of a picture of one of our gang tissue culture rooms where you could have sort of 10 trainees working together, and that's really kind of the special sauce that has made us successful over about the last 10 years. So we actually just celebrated our 10-year anniversary, which we called sort of a decade of discovery. And sort of one of the foundational aspects of what we do here at the CREM is the use of something called induced pluripotent stem cells, or iPSCs for short. So these are master stem cells that we can create from your skin cell and more recently from your blood cell to create a master stem cell line that interestingly is an exact genetic match to you. And in its undifferentiated state, it can be grown forever, and we can freeze those undifferentiated cells down, sort of share them with researchers all across the world. And importantly, they can be differentiated into virtually every cell and tissue type in the body. So that's a complicated explanation, but for you Lord of the Rings fans, it might be easier to think of iPSCs as the one cell to rule them all. Because as I noted, from these master stem cells, you can create virtually an unlimited supply of any cell or tissue type in the body. So here at the CREM, one of our claims to fame is that we don't just make one or two types of cells, we make about 25 in our state-of-the-art tissue culture rooms, which I just showed you. And what you'll notice is that each of the tissues or cell types we make is also associated with a disease that we've studied over the last 10 years or so. So for the first decade or so of my career, we worked on blood. So as Mike just noted, I'm a hematopoietic biologist. We work on sort of blood and immune cells. And so we were one of the first labs to pioneer a methodology to go from these master stem cells to unlimited numbers of blood and immune cells. And this was fantastic back in the day, because the idea is that maybe at one point we can make platelets, which are the band-aids of the blood, or red blood cells, virtual, unlimited supplies of these cells to obviate the need for blood cell donation or contamination or supply issues. But one of the things that came out of this application was this concept of disease modeling. So we worked on a lot of so-called blood-borne diseases like sickle cell disease or beta thalassemia, which are genetic disorders of the blood. And we were able to model these diseases in a dish, and were sort of very fruitful for about a decade in designing methodologies or disease models, but also countermeasures to those particular disease models. But a couple of years ago, cue the existential crisis. So I had an existential crisis, and I highly recommend to everyone, including my trainees, to have an existential crisis, because it sort of forces you to re-examine your own philosophy, but also the methodology by which you get to that particular philosophy. And for me, the question that started to keep me up at night was the following, and that was, is focusing on disease the right choice? So our center was built upon and very successful for 10 years in using disease-specific iPSCs, or these master stem cells that we created from patients with particular diseases, to recreate these so-called models in a dish. And then, of course, as scientists, what we do is we come up with countermeasures for those particular diseases, right, after the disease has manifested itself. But what I started to think about is that realistically, society as a whole and the medical schema is sort of built upon this premise of treating disease rather than the prevention of disease itself. And then for us at the CREM, one of our sort of big missions in life is to train the next generation of great scientists and clinicians. And I thought about the way that we did that. And the way that we did that is we teach our trainees to recognize anomalies, or to treat and counsel individuals only at system critical time points, when things are really hitting the fan, right? And we don't focus on things like resiliency or prevention of disease itself. So we started to think about how we could flip this script sort of on its ear. And conceptually, the big picture idea of my short talk today is this. What if instead of focusing on disease, we instead flip that script to focus on resilience, or something we refer to as dynamic resilience, which is the productive way you respond to insult or injury. So we revamped ourselves. We redesigned my own personal laboratory group within the CREM. There's about 10 different labs here. And we kind of placed ourselves at this really interesting nexus. This is a picture from our website where you can actually see some of the projects we're working on. And where this placed us was at this really interesting nexus of regenerative medicine, what we had done for about 10 years and had pioneered a lot of the methodologies for, and aging. And we saw this sort of inextricable link between the things that we were doing in the laboratory from a regenerative medicine point of view and trying to better understand the biology of aging itself. So I can't sort of begin things without the major existential question, which is a question we can't answer, which is, what is aging? It's sort of like the Matrix. You can't be told what the Matrix is, but you're experiencing it on a daily basis, right? So if you look in a textbook, right, as far as what aging is, you're going to get a definition that would take you to something like the time related deterioration of the physiological functions necessary for survival and fertility. So that's a mouthful, right? A lot of experts would simply see aging as damage accumulation. But if you ask, as they say, 10 experts of what aging is, you're going to get 10 different answers. So for us, we started to think about aging sort of from a different lens. And that was this lens of resiliency. And that maybe what aging was this loss of resiliency, loss of this ability to respond robustly to insult and injury. And then we've kind of added in more nuance lately where, yes, it probably is damage accumulation over time, but it's that fine balance between that damage accumulation and your ability to robustly and productively respond to it, right? So there were other things that allowed us to think about switching our platform, really. So going from blood cell development understanding to aging biology is sort of a big leap, but there were some other precursors that helped us make that transition. And the first is something I know, Mike, you're very familiar with, which is aging is really the strongest indicator and purveyor of future disease. If you live long enough to the 5th or 6th decade, you're certainly going to develop disease. And one of the strongest risk factor for that is simple increase of age, right? And then perhaps even more, as scientists, we never think about things from a budgetary point of view. I never know sort of the budgets that my sort of lab is sort of constructing to sort of fund our work, which is not great. But we have to think about the economics of science in and of itself, particularly in kind of the current funding climate. And one of the things that we knew and became acquainted with, which is that, rather than working on individual diseases, probably there was much better economic input and value from research that would delay aging itself. So maybe working on this concept of resilience would be worth trillions of dollars in sort of healthcare dollars based on the fact that even if you prevented the onset of disease for a short period of time, then that would actually be something that would be economically viable. Okay, so the thought outline for our talk today is sort of this new paradigm where we're going to focus on resilience, okay, rather than disease. And then also, how do we sort of mix in our stem cell-based models? Well, we know there was a paucity of human models for the discovery and validation of potential geroprotective interventions. That's a big sentence. To summarize, lots of things and lots of research has been done, elegant research that has taught us lots of things in Drosophila, in flies, in worms, in mice. But from an aging perspective, how do you develop a model that's indicative of the human condition, uses human cells, and then could validate some of these things that were coming down the pipe all over the place? A geroprotector is something that may increase your health span, the number of healthy years you have over time. So if we put something into a human or into the equation, how do we know if that has some efficacy? How do we know that there's been a change in our system? And was there a way that we could use our stem cell-based models, which come from human tissue, to try to better understand not just aging biology, but potential interventions that you might add into the equation? Okay, fantastic. So for us, we became very enamored with a particular group of people, as Mike sort of alluded to, that were really the model of this kind of dynamic resilience we're talking about, the ability to robustly respond to insult and injury. And these are centenarians. So one of my favorite people on the planet who I collaborate with and lucky enough to for a long time now is Dr. Tom Pearls, who is the co-director of the New England Centenarian Study. I'll show you his picture in a second. And he basically has this famous saying of the older you get, the healthier you've been. And this is something that's exemplified with centenarians, people that live to 100 or better. A lot of the people that we've studied in our cohort are 105 or better, which is even more rare. And so centenarians display something we refer to as exceptional longevity, the ability to sort of live robustly without the onset of disease or disarray. And as I've noted, they really do display and are a model for this concept of dynamic resilience. And they show something we talk about lifespan and health span. You know, we're not working in the laboratory to increase lifespan. We want to increase health span, the number of years without any kind of diminution, where we can delay cognitive decline and the onset of aging-related disease so that you can continue to do things like break the skydiving world record at 104 or vote and do all the things of daily living that you've been accustomed to, right? It's no fun if you're 100 years old but not able to sort of live life as vitally as you've always done it. So this is Tom. And one of the things that this collaboration allowed us to do here at BU was to gain access to these elite people who are very rare, and they're, you know, such amenable, amazing people. They're very sort of happy to be involved in some of these studies. So what we did was we went through sort of a clinical characterization and cognitive assessment. Tom's group, Stacey Anderson's group here at Boston University, they do this sort of elegant phenotyping of these particular individuals. So there's different flavors of centenarians, which we can talk about later, and there's different levels of cognitive stability and people that are amazing even within the centenarian brand itself. And what we did is we collected blood samples. So here at the CREM, we had pioneered the methodology to make those master stem cells, those iPSCs I told you about a couple of minutes ago, from the peripheral blood mononuclear cells. So basically from about a teaspoonful of blood, that's how we can make these custom stem cell lines from these highly specialized individuals. But what we also did as a hematopoietic biologist, I was also interested in the underlying hematopoietic and immune constitution of these individuals. So it's definitely not a stretch to basically assume that probably these individuals who are resistant or resilient against aging-related disease probably had elite immune systems. So we did two things. We used a tiny bit of that blood to go through this process called reprogramming, which is how we make these matched, genetically identical custom IPS lines. We also performed something called site-seq, which is kind of a complicated nomenclature, but simply what that means is we perform combinatorial single-cell sequencing, so mapping the transcriptional profile of individual immune and blood cells within these specialized individuals, but also using a little bit of a oligo tag and antibody where we can get simultaneous transcriptional and protein level information all at single cell resolution. And then the other cool thing that we did, we love FACS profiling, which is a way to interrogate, you know, millions of cells at the protein level in a very different way. We use spectral, something called, it's a big word, spectral flow cytometry in coordination with this other site-seq technology to really interrogate at very high resolution the immune and blood cell type landscape of these really elite individuals. So this is one of the coolest things. We had centenarians in our cohorts who had actually survived both the 1918 Spanish flu, which killed an amazing number of people, but also had COVID a couple of times and just shook it off and kept on moving. So I think one of my biggest revelations when I began to study these populations was it's not so much that these individuals are indestructible and they don't get disease or they don't get the Spanish flu or COVID. It's how they bounce back from these insults. They respond so much more robustly than normal individuals. That's really what we wanted to study. So it's their ability to sort of show this dynamic resilience when there is an immune insult, that they can overcome that in a very elite way. We also found in this paper, and we're going to discuss it more later, that there were separate and distinct signatures for aging and extreme longevity. So if you look at your immune system and your immune cells, there are definite age-related changes that are common amongst everyone, as far as your cells becoming more myeloid biased, things of that nature. but we also found a separate and distinct signature using these methodologies of extreme longevity, of these elite individuals that had reached these astounding ages. So for us, this really did provide this blueprint for trying to understand this new concept of resiliency and how we might sort of, take all this information and increase health span in, a non-elite individual. Okay, so the last part of my talk is how do we put all of this together? So how do we take all this information, take the stem cells that we made, put it together to come up with this human-based model to study aging and maybe these interventions that we want to know if they have efficacy or not. So the first thing that we actually did was we built the world's largest bank of these master iPS cells from centenarians and their offspring. So longevity is very genetically penetrant. So not only did we actually make iPS cells from these elite individuals, generally 105 or better, but also from their offspring, because we thought maybe these middle-aged individuals would also have very sort of informative lives and genetics that we could study. We did this from both males and females. Interestingly, women live longer, but 85% of centenarians are female, but we had a nice mix of male and female centenarians. And what's nice is that we didn't just create these master stem cell lines that we could share with the world and research in our laboratory, but we could connect the data that Tom and Stacy here in the New England Centenarian study had collected to connect this really amazing phenotypic data about these specialized individuals with their coordinate stem cell line in a dish. So that was absolutely fantastic. So what does this look like? It's kind of our trade here at the Creme. We go through this really sort of elite phenotyping of these master stem cells that are made from these individuals. And again, when we make the line in its undifferentiated state, it can grow forever. So that's a one-time thing. And then we have an unlimited supply of material. We look at the karyotype of these individuals. One of the cool things that we found, and we're still trying to sort of get our heads around, is that in the male centenarians, about 30% of those males had something called mosaic loss of Y, where they lost the expression of their Y chromosome, which generally is sort of correlated with increased rates of cancer and heart disease. But we also think it could be protective, particularly in these really elite specialized male centenarians. We also ran teratoma assays, which just kind of showed the functionality of these iPS cells, that if you just put them into a plug, a matrigel plug, and grew them, you could create all the cell types and all the germ layers in a particular sort of situation, which is important as far as dictating whether or not these cells are something called pluripotent or have the ability to create any cell or tissue type from that master stem cell line. And then lastly, one of the cool things that we did is from the peripheral blood cells that we used to make the iPS cells, we also used biological aging clocks. So I have a postdoc in my laboratory. His name is Nick Scavier, and we call him the clockologist. So he's an expert and did his PhD on the construction and application of biological aging clocks. And so we ran a series of biological aging clocks, including one that was specifically trained on centenarian data. And what we showed is something you'd expect. If you look at the chronological age, the age that it says these individuals were on their birth certificate versus their biological or molecular age, what their body or their physiology was saying, they were 20 or 30 years different, right? So these people were much more, as you'd expect, much more biologically young than their chronological age. And so what we did is we used these data to stratify which of these specialized individuals we then take forward to try to create this model of resiliency in a dish from the stem cells, right? So if you look in the bottom right-hand corner, one of our specialties here in the Creme is we can take these master stem cells and differentiate them into multiple cell types. So there's a little bit of eye candy for you where we can actually make neurons, an unlimited supply of cortical neurons, basically the brain cells from these individuals in a dish. So these are basically equivalent to the 100-year-old neurons that you'd see in these individuals, but we can plate them in a dish and run lots of assays. Okay, so how does that work? And this part of the story is basically iPSC-based or master stem cell modeling of this resiliency in a dish for us to try to understand what are the true signatures of longevity that these specialized centenarians are displaying, right? So how do you create that model in a dish that would allow us to apply it to all these interesting biological questions we have? So what does that model look like? Here we go. So what we do is we take these master stem cells, these iPSCs, from centenarians, those with exceptional longevity, but also controls, people that we knew had no history of longevity, right, as a control for the system. And then because of the inherent flexibility of iPS cells, we could differentiate them into virtually any cell type in the body. And we chose 4 that we knew had aging impact. These are some of the tissues that as you age are strongly impacted by that aging process itself, including the brain. And then the idea would be we would apply a series of mechanistically distinct stressors. So what does that mean? You experience stress every day of your life. Maybe you take a plane flight and you're bombarded by x-rays, things of that nature. You're experiencing stressors on a daily basis. In a dish, could we recapitulate this type of stress in the system to see how these specialized individuals who we know respond in real life very effectively to these stress and insults, could we recapitulate that type of dynamic resiliency in a dish? And then, of course, we use our fancy molecular tools where we can do transcriptional and epigenetic profiling on these cells to see what was the molecular signature of this resilience. But really importantly, and I think one of the critical things we're working on now that I think is absolutely paramount as we go forward, is it's not enough for us to have these molecular signatures of maybe how their blood system is different or maybe how the cells respond in a dish, but we needed actual functional readouts to see how the cells might perform differently in a dish. That was something that we felt was essential to be instructive in the process. Okay, so let's perform one of these experiments together. So here we go. So here's the iPS cells that we have from controls. These are 30 to 50-year-old individuals with no history of longevity. And then we have a series of our elite centenarians, which we stratified the best of the best for these particular modeling in a dish assays. What you'll notice is that we actually don't give the actual age of the centenarians because they're so rare. The age itself is an identifying characteristic. So we have to give a range because of the fact that these people are so exceptional and so rare, you'd be able to identify them just by their age alone, right? We have some of the oldest people in the United States that are part of this cohort. So we took these master stem cells, as I showed you before. We go through a specialized differentiation process to create neurons or brain cells in a dish. And then we add a stressor in. In this case, we're adding in something called dapsogardin. That's a complicated term. This is just a global ER endoplasmic reticulum stressor that creates an issue with protein processing control. So if you go through dementia or cognitive decline, you probably have issues with your protein processing control and how you deal with aberrational proteins in the particular system. So here we're actually creating stress in that system to try to exacerbate issues with protein processing control, okay? So it's one particular type of mechanistic stressor. And then, of course, we look at our fancy tools at the end to see what the results might be and whether or not the specialized individuals who display exceptional longevity maybe respond differently. Okay, so what's the $100,000 question? What did we find? And this is in the new paper. So this is something called a volcano plot where we can actually look at the molecular signatures of resiliency based on that transcriptional profiling I talked about a second ago. And what we found specifically in the neurons from individuals with exceptional longevity, when we stressed them, as I noted, was that you had increases in neuronal functionality. You had signatures of protection against things like Alzheimer's disease and related dementias. And really cool, my favorite result is that you saw signatures of neurogenesis or the birth of new neurons. So it seemed as if these specialized individuals could regenerate their existing neurons or perhaps even birth new ones. And this sounds crazy. It sounds a little heretical, but there's actually a new paper that just came out in Nature, Mike, you've probably seen it, where they actually looked at the brain tissue of elite cognitive centenarian superagers, and they saw actual evidence of new neuronal growth there. So the old adage of maybe you have as many brain cells you're ever going to have and you don't create new ones, maybe that can actually be altered in particular situations. So really cool signatures in these individuals post-stress of the birth or creation of new neurons in a dish. Second, when we added that particular stressor, that global ER stressor, what we saw was really cool. So this is a heat map. Blue is off, red is on or higher. And in the naive state, so this is looking at the exceptional longevity individuals without anything added, with just the vehicle alone, you see a lot of blue, right? But then when you add in the stressor, you see upregulation of many different genes. So one of the striking things that we saw, which has now become a commonality across lots of different ways in which we're examining these individuals, is if you look in the naive state without the stress, of the individual neurons from individuals with exceptional longevity versus the non-EL versions, you just see a globally quieter landscape. It's almost as if there's less noise in the system. So at the basal layer, before the induction of any stress, these individuals and their neurons seem to be walking around in a much quieter state, less entropy, less noise in the system. But they reserve this capacity once you induce a stressor or an insult where they robustly upregulate the quality control mechanisms to deal with that particular stressor. So off or very low in the basal state, which is probably more fruitful energetically, but when you need to, they can fruitfully upregulate mechanisms very efficiently to deal with these types of stressors. So that's also really cool. And then lastly, the question you might ask yourself is, well, is all of this just an aberration of this model these guys have created in a dish? This is just some sort of stem cell magic and it's not really applicable to other systems or in real life scenarios. What we did is we had a signature of resilience now. We had a molecular signature of genes that we thought, when upregulated, were a signature of this dynamic resilience. We then took that signature and decided to compare it to other databases in real life sort of situations. So one of the things that we compared it to is something called the late onset Alzheimer's disease database. And when we compared the signature that we saw in our dish, it was more closely related to age-matched individuals who did not have Alzheimer's disease and related dementia. So more closely associated with those individuals at a same age who didn't develop disease. We also compared the same signature to cancer databases, and we saw that the resiliency signature that we found in the dish was more closely related to cancer resistance, right? So that's also cool. So we think that this signature might be universal across systems, tissues, and maybe individuals. So that's still to be determined, but that's the next phase of what we're doing. Now, the last thing that I'll show you has to do with what we said before was super important. So this is great. We have these molecular signatures, lots of transcriptional regulation, lots of really interesting genes that come up when you stress exceptional longevity neurons. But what about those functional assays? Do these cells that seem to be better molecularly from the centenarians, do they perform better in a dish, right? So one of the first things that we did is in collaboration with Vadim Gladishev's lab from Harvard, we used his transcriptional aging clocks to plug in our particular data. So one of the first things that we saw on the left, sort of here in pink versus blue, when we added in that stressor I talked about before, that thapsigargan that dysregulates endoplasmic reticulum control of protein processing, we saw that when you added in that particular stressor, really interestingly, if you look at the transcriptional age of those cells, they got older. So that's kind of cool. Maybe that means something, maybe it doesn't, but it suggests that that stressor increases the age of the cells in a dish. And then conversely, without any stress, if you look at control individuals versus those that display exceptional longevity, these are neurons from those individuals. It seems, if you look at the mortality transcriptional age in the individuals with exceptional longevity, that seems to be much younger. And then here are the functional assays. So we've kind of gone through a number of functional assays. The assays that we're using now in the laboratory going forward are things that are directly connected to the hallmarks of aging themselves, things like mitochondrial activity. And again, this is one of the coolest things that we saw. When we looked at the mitochondrial activity of the neurons of the centenarians, the first thing that you'd think to yourself, maybe teleologically, is, man, if you meet these people, they're incredibly robust, they're energetic, they must have high mitochondrial activity. It turns out, and this was surprising to us, that the exact opposite is true. So if you look at non-EL neurons from a mitochondrial activity point of view, this is something called membrane potential, versus those with exceptional longevity, again, in the basal states, the mitochondria, your powerhouses of the cells of individuals with exceptional longevity, their resting basal state is way lower than a normal individual without this history of exceptional longevity. And so at first we were a little bit confused by this, but then we actually looked to our friends that study other long-lived mammals, like the naked mole rat, which, unlike a normal rat that lives a year or two, these naked mole rats can live 30 or 40 years. Or how about the bullhead whale, which is the longest lived mammal that can live 200 years or more? It turns out that if you look at their mitochondria, they have this conserved sort of application of lowering their bioenergetics as a potential longevity strategy. So maybe the less energy you use in your basal state the more conserved your energy is, and that is a longevity strategy that's shared across species. Well, what I will say, and the data is not here, but it's in the paper, is that when you then run an assay like Seahorse to look at the dynamic range of how these mitochondria react, they can still sort of upregulate all of these processes in a normal way. It's just at that basal state, everything is much lower. And then lastly, and maybe more cool from an eye candy point of view, we're also able, using live calcium imaging, to look at these individual neurons in a dish and then multimerize these using various R scripts in a heat map type of way to look at the synaptic functionality and capability of these cells in a dish. And this is what it looks like. This is a movie where you can actually see the individual neurons firing in a dish. We can capture this using high resolution methodologies and then put these into heat maps like this. So these are neurons on the left from somebody with a genetic epileptic disorder. You have the non-EL controls in the middle. These are people with no history of longevity. And then look at the difference that you see in the synaptic activity of individuals with exceptional longevity. It's much more ordered. There's less noise in the system. It's just better and sort of more functional and more robust without this sort of entropy in the background kind of basic noise. So again, kind of holding to this sort of more elite signaling, and in the basal state, a sort of a quieter, more ordered landscape. So my last slide is the following, okay? So unfortunately, based on genetics and the lottery of genetics itself, the likelihood of you being a centenarian is low, right? These are elite, very rare individuals. So the next phase of what we're doing is, how do we make you more centenarian-like, right? So now that we understand what might be these human signatures of longevity, can we then test particular individuals as far as whether or not a particular geroprotector or an intervention might work? So it works a little bit something like this, and this is something that we've repurposed from our disease models, and we call it a clinical trial in a test tube. So if you have an individual with a particular disease, we can create that model of the disease in a dish and then come up with a countermeasure for that disease. We've taken that a step further where we can make you a custom stem cell line. Maybe you're completely healthy, but you want to understand how you can increase your healthful resilience and health span. We can also look at potential geroprotectors, things like rapamycin or maybe GLP-1 peptides. How do these potential geroprotectors that may increase your health span, how do they work in you on a personalized level? And can we actually come up with particular regimes that will work specifically for you based on this human model of development and disease that we've developed? And that's sort of the next step of what we're currently working on in the lab. Now, I'll stop there and basically say, literally everything we do at the CREM is based on this teamwork approach. The people in my laboratory, Todd, Elise, Sam, my students, Natalie, Nick, my clockologist, are absolutely amazing. It's the trainees here that really are the special sauce that have made the CREM where it is today. Love my collaborations that I have with Tom, Stacy, Brad, and everyone at BUSM. Also, Paolo Saviciani at Tufts, my collaborators at Harvard, and obviously the support that comes from the NIH and the National Institute of Aging, absolutely essential. And I will stop there.
That was fantastic. So I have to give a few quick shout outs first. So, and I usually do this at the end, but I want people to know about it from the jump. So you have your own YouTube channel called The Reprogram. So if people want to check that out, I'd head over there, subscribe, and check out George's videos. Two is, can people join your lab? Like, because there may be students that are listening and that, I think your lab is great. If there are students who are interested in aging and longevity, it would be a great place to. So are there students that could join your lab?
Yeah, so we literally have 70 trainees here. It's really run by the trainees who are the lifeblood of the CREM. All of the laboratories, including my own, are always bringing people on board. And even if people that are in Boston want to see what we do up front, we have right now, we're having a stem cell course where literally anybody in the world can come to learn how to do what we do as far as manipulation and creation of these cells. And we're always onboarding trainees and are looking for fantastic people who want to sort of live by the mission of what we do here at the CREM. So definitely love to have people come visit. Definitely always looking for students and trainees, and that is the lifeblood of what we do.
And then third is, I don't know how BU's rules are for people trying to support your lab with donations, or even if that's a thing. Tufts was very rigid about that. You can't, but is there a way, does BU, are they more permissible with being able to fund scientists directly?
Yeah, so the development office here is great. If you go to our CREM website, so if you just look up CREM BU, we actually have buttons that allow for a direct donation there. And obviously, as you know, the funding landscape of science is a bit of a disaster currently. And so obviously anything people can do from a philanthropic point of view or even from a general public point of view to support research in general, I think is an amazing thing.
Fantastic. All right. So the challenge now for me is you have three components that I'm extremely interested in, and I always go to the most interesting first. So now I have to mentally juggle which of those three things I find the most interesting to start off with questions. So I think maybe starting from the back. So, or maybe not the back, from the end of your presentation. So you calcium signaling dynamics and membrane potential. So I'm sure you're familiar with Michael Levin's work, where bioelectrical signaling during aging, starts to get distorted. So the signals going to the cells, basically you have, older cells because the signals are messed up, right? So how do you foresee, for people who missed it, because you're the co-founder, co-director of the Center for Regenerative Medicine at BU. So how do you foresee, because you're studying membrane potential and calcium signaling dynamics, how do you foresee studying that in centenarians and then interventions that could tweak bioelectric signaling to potentially improve actually, at the personalized level, people who are sending you their cells and then you could study it. So how do you foresee the future of regenerative medicine using bioelectrical signaling?
So I think it's a super interesting field and obviously emerging like much of the things are in aging and longevity. The other thing I would say is a lot of the things that I described sound super high tech as far as, we can make a brain organoid, which is sort of a mini brain in a dish. All these things sound super cool and, technologically wonderful, but they are cave person tools, right? They're still very crude models of things. And although we are very specialized in what we do here, we're always looking for the next thing that's going to take our models to the next level, that are going to more faithfully recapitulate who does it best, which is Mother Nature. We can never be as good as that, but we look at sort of the way that nature goes through development, and we simply try to recapitulate some of that in a dish. So things like, bioengineering or, matrix models, we're very primitive there. And I think bioelectricity also fits into sort of that category. So what's really cool is that I'm going to put on my developmental biologist hat for a second. We at heart here at the CREM are developmental biologists. And as I said, we look to the developing human embryo to try to, inform the models that we develop. And that's how we figured out a lot of these differentiation protocols. So I think bioelectricity is really sort of one of the hidden layers, right, in developmental biology. And there's these really cool experiments. I love planarian. If I could go back, if I was a student, I would go to a planarian lab because it's such an amazing model and way easier and less time consuming than some of the things that we do. But there were these amazing bioelectricity models where you could change the development. You could create a planarian with two heads, or maybe in higher order organisms, you could change sort of the bioelectricity to sort of maybe reinitiate the things that we're trying to do here as a Center for Regenerative Medicine, which is maybe regrow a tail in a species that doesn't have that, right? So for sure, we think that bioelectricity will regulate things like calcium entry. We're really interested in chromatin state. We think epigenetic changes are what maybe drive aging. So we don't just use those clocks to try to understand maybe what the age of our models are, but we think it's actually causal. That hasn't been directly proven yet. But for sure, we're really interested in sort of bioelectricity as far as looking at one of the fate choices that we, perform every day, which is, do you proliferate or do you differentiate, right? So I think what's more murky, and I think we need more of a spotlight on, is the concept of memory, that you have some sort of anatomical memory that kind of illustrates morphogenic intelligence, the way that something develops, right? So I think that's a bit confusing for me, but I think for sure, we're just scratching the surface with the different methodologies. And for us, we try to take all comers. We try to be early adopters. We were some of the earliest adopters with spatial omics and got our butts handed to us because that's what's supposed to happen. But we're really interested in bioelectricity as a whole. We think it's a big player. It's something that we haven't really explored, but we definitely be interested in doing. I think it has a tremendous upside in future, particularly in developmental and regenerative biology.
Nice. So if you partially depolarized, so in looking, so that your membrane potential studies with the mitochondria surprised me, right? So if you partially depolarize a controlled mitochondria to the same membrane potential as in the centenarians, would their oxygen consumption and other measures that the seahorse measures, would they be similar, or would the controlled mitochondria now be actually dysfunctional and somehow it's not just the membrane potential in the centenarian mitochondria, it's something else that's also contributing?
We talk, Mike, that is the perfect question, and we talk about this every day in lab, right? So one of the questions, and one of my grad students has to contend with it. This is one of her biological questions: is what is a longevity signature, right? So people would normally think you need to add something into the equation to, influence your mitochondria to have more bioenergetics, to, have more turnover, have more membrane potential. And so, you would have this supercharged mitochondria, and that's what you want. I think what our data shows, and some of the long-lived mammals say, is maybe that's not the methodology. Maybe the longevity signature is you actually, in the basal state, keep these things at a very low resting level, but maintain that ability to respond when you need to. So one of the questions we're asking now is we're using GLP-1 peptides, for example, or we like this PGC1 alpha sort of additive, which is known. These are things that are known to be involved in mitochondrial dynamics. And the question for us is when we add these things into an equation into a person like you or I that's not a centenarian, what happens exactly like you noted with the mitochondria? Does that increase the membrane potential, and is that good? Or does it then create this sort of basal state that's more like what we see in the centenarians? And is that really what you want to get to? So what we're struggling with now, and we don't know the answer, is what is the right outcome that we want to sort of import into a normal person that would be indicative of you living a longer period of time, right? So that's the challenge. How do you do something today that way down the line is going to have this additive, beneficial effect on your health span? Is it turned up mitochondria? Is it turned down mitochondria? We think it might be turned down. Everything turned down until when you actually need it.
Could the turning down of the membrane potential in centenarian mitochondria, could that be a compensatory mechanism to limit oxidative stress production?
Totally. I forgot to mention that. Yes. So that's what we see, right? So why would you have that? So exactly as you're saying, in the resting states, if everything is lower, even your protein processing control seems to be very low, then you're going to have better sort of, stress management, better, oxidative capacity, all of the dynamics of the things that you have in reserve. You probably have more of a dynamic range. And so what I didn't show the data from is that when you then run Seahorse, which again is a fancy tool to actually look at these things like, oxidative metabolism, things of that nature, you still retain this robust capacity. So there's no difference between the neurons once they're highly activated. It's just that in the resting state, everything is way lower, less energetics, maybe more efficient respiration, things of that nature. And we do see signatures of more efficiency in the basal states, but maintaining that dynamic range to respond when you need to.
Nice. What are your thoughts on comparing the centenarians who had an older versus younger biological age, right? Because there's going to be a distribution there. So if you compare within that category and then look at, so I'm obsessed with the bioelectricity approach. I get the epigenetics and I get reprogramming, but the signals coming from the top can drive everything independent of the genes. So if you compared older, quote unquote, older versus younger, at least by the epigenetics, would the predicted older, would they have a either lower or higher or different membrane potential? And then if you reset, actually, not just biological age, I'm sure within the sentence, sorry, George. So so I'm sure you have mortality data in the centenarians, right? Yeah. So if you knew how long, centenarians who live longer and centenarians who live shorter, and then compared membrane potential in the iPSC-derived neurons or other cell types that you get from them, I wonder if they'd be different. And then can you take iPSC-derived neurons or any other cell types in the shorter lived ones, and if you hyperpolarize or not hyperpolarize, but increase membrane potential, do you get some of a restoration of their functional state? So I guess these are plans to do, or yeah.
Such good questions and are exactly our thing. So what I would note is a couple of things, and I learn something on a daily basis, as I know you do, and I have a voracious appetite to understand things. And centenarians are this wealth of information. So what's really interesting is that in the populations that we study, even though these people have exceptional longevity, generally the mortality rate is 50%, right? So what's awesome is that we can make these IPS lines that'll, exist in perpetuity and kind of go back with things. Because I always say this, we're stem cell biologists, so we always work on worst or best case scenario. It's generally worst case scenario, the worst disease, the worst phenotype of that particular disease. Then we create the line from, and then it's hard to create a disease model. So you want the worst of the worst, and then that's what you start with. We did the opposite here, which is based on those biological aging clocks, we took the most elite of the elite. So we had people that we would call super centenarians that are 110 or better. And then when we looked at the phenotypic data, as far as frailty data, as far as They do these amazing phenotypic characterizations as far as walking speed, grip strength. They send sort of the centenarians a kit that allows them to gather all of this wonderful phenotypic data over about 2 days. And so, really interestingly, we ran these molecular biological aging clocks. In the people that we saw the biggest differences between their biological and chronological age, sure enough, they were the people that didn't have cognitive decline. They performed the best on the cognitive tests. They were the people walking around, living independently. So there was really interestingly this correlation with better biological age or younger biological age and functionality. As far as people always ask me, well, how do you control these experiments? There's no perfect control for a 100-year-old, right? But what we would do is for some of the offspring, right, where maybe the genetics there are about 30% penetrant, we would collect the sample from their spouses and then we plug those into the clock. And you'd see that person would either have a terrible biological age or something that was really close to their chronological age. And then you'd see the people that we would predict to be centenarians, we saw a bifurcation in those individuals. So the offspring, people that maybe got the magical genetics versus different, we could see this huge trajectory difference in the clocks based on maybe they had the underlying genetics, right? So I think these tools for us have been super useful in stratifying who are the people we want to study. But as you noted, the first thing that we did is we picked 10 of the people that were the superhuman, best differences in biological age, most functionally competent. And now, as you noted, we're going back and we're looking at a frailty index where we're taking the 10% worst performers in biological aging clock, but also in phenotypic measures and comparing that to the 10 best and trying to not only see those differences, but then if we're testing geroprotectors, can you make one like the other? Or in a normal person, can you import our resiliency signature into a normal person and see the same type of dynamic changes, both molecularly, but also in these functional assays? Do the cells perform better when you say put in the 65 resiliency genes that we know are expressed in a centenarian when you stress their cells?
Nice. So technically, if I understand correctly, you could take cells from the oldest old and then use a variety of quote unquote longevity-based interventions and even restoring bioelectric signaling or increasing memory potential as a part of that. And then in a head-to-head matchup, which is best for restoring a more youthful state, rejuvenated state. No, oh, fantastic. Holy cow,
Yeah.
Joey.
And then you could go even deeper there, where one of the other things that we're doing is making chimeroids or chimeric models, where maybe you take somebody with Alzheimer's disease. So we have models in which we have neurons with somebody from Alzheimer's disease. What if you create an organoid or a bi or triculture model where you culture neurons or astrocytes or microglia from one or the other together, or from a good performer and a bad performer, and then you look at the outcome? Can you have compensatory mechanisms that will adapt the ADRD cells or vice versa? Do you see a disease process, or do you see an increased signature of resiliency to really test whether or not these cells can overcome these bad outcomes, right? So there's lots of really cool mix and match things that we can actually do to try to understand the performance of how these cells work, both from a centenarian elite point of view, but also from standard individuals, how they might respond in similar situations.
The chimera is an interesting concept because just going back to Levin's work, they find that, if enough cells with the message, with the bioelectric signaling message, they then go and convince all of their cellular friends to follow that message. So in the chimeras, I wonder, like, if you hit it with the right bioelectric signal, it doesn't matter if you've got an AD, ApoE, E4, E4 phenotype, right? The cells are derived from that. If you have enough quote unquote good cells, heteroplasmy, right? Potentially you could drive the whole thing back to the functional state.
Totally. And like, that's why I see this really intricate connection between what we studied forever, regenerative medicine, and now our new field of study, which is, aging biology, because the reprogramming process in and of itself shows you that the entire system is malleable, that you could basically put signals at the end, middle, and maybe upload initially at first to try to change these dynamic trajectories of resilience, of disease, of proliferation, differentiation, and it's a subtle balance. But I think the take-home message, which is different now than it was maybe 10 years ago for us, is that the plasticity of these systems is robust and that everything is probably malleable, which is cool, right?
100%. Do you have plans? I don't know, maybe you're already in the process, but of building a company around this where people can give you blood, you isolate cells, immune cells, and then derive iPSCs, and then either, put them back in, or not necessarily reprogram, but identify the longevity-based interventions that would be best. At least, I mean, technically, you could do it for rejuvenating red blood cells, rejuvenating platelets, immune cells, right? And then with the iPSCs, all different cell types, you could see which specific longevity-based interventions would be best at the individual level. So, forming a company in plans, or what are the thoughts
So
there?
I always have seen myself as this tragic lifelong academic, but
Mm.
it's been increasingly difficult to ignore some of these other things. So yeah, we have some things in work. Like my last slide, we collaborate with, it's not really a company, it's sort of you may have heard of Wonder Health, which is sort of this collaborum of scientists who have come together. And our role in that is sort of the centenarian in dish, where we're trying to make these predictive models, exactly like you're saying, at the individual level, where we could use this combination of molecular and functional signatures to know you, Mike, how do you respond to rapamycin before we put it into you? In a much safer way, we could test combinations of things. How do you respond to GLP-1? What's the sort of best agonist to look at metabolic control in your particular system? So in a very personalized way, over this collective, we're trying to do that. The other thing I would say, and kudos to the NIH, is that we are part of, we're lucky to be part of the Longevity Consortium. So within the Longevity Consortium, one of my other hero scientists is Fred Rusty Gage, who I'd always, like in my stem cell class, I use his slides. And now I get to collaborate directly with him on a project. And our project is exactly like what you're describing. So from an academic point of view, even the NIA wants to know, hey, we need a model, a human-based model, to kind of predict and understand how these particular interventions, there's a lot of hand waving going on, but can you show us some real evidence that says if you put on board this particular intervention in a human system, how does it react? So I'm a huge Rich Miller fan. I love the interventions testing program. You cannot do better science, but that's in a mouse, right? And what's really interesting is that you see the sex bias in mice. You see very differential effects that sometimes the crossover into humans, a mouse is not a human and a human is not a mouse. So we think it fits well into this equation of more understanding is better. And particularly if you can do that in a human system that's connected to the genetics of the individual, then maybe that's the way to go.
So it sounds like staying in the academic, on the academic road, and not having I'm not saying jump out of academia. Both can be done. So yeah. Wait, so the X Prize, are you familiar with the X Prize? This may
Sure, yeah.
have,
So we collaborate with Nick Shork, who's heavily involved with the X Prize itself, for sure.
yeah. So, I mean, that may be a good funding source if that wasn't already on the radar, because I'd be shocked that they wouldn't be interested in funding, a personalized approach for discovering your own, longevity-based intervention strategy. So that may help in terms of funding. So
Yeah, I appreciate that. Always interested in funding. I think one of the cool things we've done at the CREM is that we operate a system where we have lots of patents on like the way that we make blood. We have that patented. We don't commercialize that stuff. We let everybody use it, but we allow everyone to use it. We have something that we refer to as open source biology, which is the unbiased sharing of everything we have. Whether that's you in Alaska and you want to use our lines, we'll send you them for free. Anybody that wants any of these lines, we'll send you them. But then we also collaborate with large companies where we have this sort of same standard research agreements where a company wants to understand how their particular drug works. Sure, we'll do that, but we don't want to own your thing, but you can't own our stuff either. So we kind of still operate that in a grant type situation, even though we have active collaborations with commercial companies.
the challenge, just to push back a little bit there, the challenge there is, and I hear you, it's like, here's all of our data. Anybody can reproduce this. If you guys want to make a company based on my work, we're happy to help out, right? But the challenge there is, like, I think so many of the longevity field, whether it's in academia or even in the lay public, if you ask them about, like, how do you foresee, quote unquote, longevity and extending health and lifespan, they'll say, oh, NAD, or, oh, it's epigenetic reprogramming. Sure, that may be small components of it, but the stuff you're doing is so cutting edge. And I don't see any other labs with this access to centenarian data and this deep insight. So it's almost like if you don't do it, it may not get done for decades, if even that long, so.
So to give you a hard example, I loved the piece that you actually did on tryptophan. I know we have a mutual interest in tryptophan. In my past life in blood, we worked on the aryl hydrocarbon receptor and basically showed that it was kind of instrumental in immune cell and blood cell formation. So not just bad stuff, it actually did some good things as well. So in the tryptophan cascade, in our co-collaborator, Paolo Sebastiani from Tufts, is that can you use the system to look at kind of the mitochondria or the tryptophan dynamics metabolic differences that you actually sort of see in people. And we talked about, the tryptophan kynurenine ratio and things of that nature. So my really brilliant grad student, Elise, basically took a normal person's iPS cells, differentiated those into neurons, added an inflammatory response with something called interferon gamma for your listeners. You see a massive upregulation of all these inflammatory markers, but then showed if you inhibited the breakdown of tryptophan, you could quench very specifically that inflammatory response. And then if you look from line to line, if you look from males to females, we could show that there's a differential there as well. So this concept of this personalized way of how you might respond, even from a biomarker point of view, now we're actually looking into how do we look at that ratio in a dish, right? To try to understand, just from the jump, can we recapitulate sort of how these dynamics might be happening in a dish? And then can we actually put on board countermeasures? So like IDO1, which, catalyzes that reaction, we can use an IDO1 inhibitor to show that we can prevent sort of that inflammatory cascade from happening. So I think it has a lot of promise, not only to show functionality of these particular geroprotectors, but to show the individual responses based on a person's genetics that you actually see. So again, coming up with this tailor-made way in which we could tell you the dosing, the toxicity, the sort of range of, dynamic response you might have to X or Y, or how a combination of things might work in your particular system. And we don't need you. You could be out doing whatever you're doing, and we could be in the lab figuring out in your genetic background exactly how these things would work.
Nice. That's great. All right, perfect. So tryptophan and kynurenine, for people who aren't familiar, and this may seem like esoteric, but the fact is that there's a growing body of literature that links IDO1 activation and an increased kynurenine to tryptophan ratio with all kinds of bad outcomes, including neurodegeneration. So just for people for a little bit more background, I didn't care about that at all until LLFS, we saw increase during age, KTR. But then for those who don't know, New England centenarian study, sure, the kynurenine to tryptophan ratio increases during aging. But then I asked Stefano, I was like, well, how important is that? So what? It increases during aging. Lots of stuff increases during aging. So then we looked at age-related changes for all of the other metabolites that change during aging. And I mean, if you have one thing that's associated with another, I don't know. I don't care. But when you have one thing that's associated with 50 things, probably important, right? So, and the direction of the change for that big group of metabolites, that the kynurenine tryptophan ratio. So if the metabolite increases during aging, it was inversely associated with KTR. Or if that metabolite decreases during aging, having a low KTR was associated with higher levels. So now I'm like, all right, what's going on there? So then, all right, so wait, what I'm getting at there is, in centenarians, it could be interesting to, if there are SNPs in IDO1 that would lead to less activation and a lower KTR, and then you'd be able to directly investigate that in the iPSCs, where, do they have a different metabolic profile inside the neurons because they've got the SNP, so yeah.
That's the exact experiment. So basically Paula has these variants, right? And so now what we're doing is we're kind of looking back in the IPS library, just because that would be the easiest thing to do, and looking at the genetic background for this rare mutation in IDO1 that no one knows what it does, but it's in a person that is like exceptionally long lived and very healthy. And so now one of the things we're doing is finding the actual IPS lines that may actually have these variants, and then doing exactly what you said, running the same profiling I just showed you, and see if just in the basal level, are there dynamic differences in the things we're seeing, right? And so that's like super straightforward for us to do, and I think would be really cool. And then connects, how beautiful is it when you can connect the genetics to sort of the molecular and functional outcome in something that you could then alter the dynamics of the system, right? So I think it's really, really exciting stuff.
Yeah, and I know you did the transcriptomics, and the iPSCs and even the single cell immune cells from centenarians and controls. But what about metabolomics, right? So really getting a functional readout by looking at intracellular metabolomics, plans to do that?
There's plans to do that. People always talk to us about proteomics. It's just, there's all this firepower and all the expense that's attached to the firepower. Obviously, Paula, Stefano, yourself, a lot of people I think you guys are in the field du jour, which is metabolomics, I think are going to guide us, through everything. We would love to look in our systems. We haven't done that yet. It's a reductionist system, so it'd be very easy to sort of make an unlimited amount of sample, to purify it in whatever way you'd want, and then to run some of these assays. And like I said, for us, now I'm going to put on my molecular biologist hat. We don't do protein biochemistry or metabolomics or any of that stuff. So even looking at the ratio of these things in a dish for us, even though this is a state-of-the-art laboratory, is something we really need to think about. So we definitely need to talk about that more as our collaboration expands.
Yeah, definitely. All right. So the thing about transcriptomics is it's not just which transcripts are expressed, it's also the system being unable to induce expression. So have you looked at which transcripts weren't there in the iPSCs versus or iPSCs derived neurons versus which were there in the younger ages?
So we haven't looked at that, but we have another paper, and I'll send you the draft of this paper where so for us, one of the drawbacks for people that know a little bit about what we do, really interestingly, 5 years ago or 10 years ago, if you said, hey, I'm going to use iPS cells to model neurodegenerative diseases, people would say, you can't do that. You're trying to model a disease that takes 5 or 6 decades to even begin to manifest itself. Your cells are fetal and embryonic in nature, and that's the drawback. Because of the reprogramming process, the cells that we make have a signature that's younger, right? So for some reason, we've now been able to get past that hurdle of people will accept if we're modeling ALS, Parkinson's, sure, you can do that. You can do drug discovery with an iPS cell for neurodegenerative things. Now the barrier is aging. If you say we're going to use iPS cells to study aging, people are like, you can't do that. This cell is rejuvenated already. It's young through the reprogramming process. How are you going to look at aging related characteristics? Because if you look, and we've done this, if you go through the reprogramming process, if we took your blood, we reprogrammed it, you would actually see your epigenetic clock going back to 0, like nascent, right? It's super cool to look at it in a different way. So now what we're trying to do is reinstill the transcriptomic and epigenetic changes that lead to the age accumulation, trying to model that aging in a dish. And the way that we're doing it first is we're using disease as a driver of this accumulating transcriptomic and epigenetic signature, right? And what we're finding is when we look at some of our disease models that have hallmarks of aging, like fibrosis, autophagy issues, protein processing control issues, for the first time in some of our organoids, we can see huge ages that you've never seen before in an iPS model, because if you look at that in a clock, it's always very young, right? So then now when we go back and we try to look at what's different, it's super complicated. So, and my clockologist is looking at this, we're trying to do exactly what you're saying. What gets turned on, what gets turned off that allow for these aging and resiliency cues to work? And it seems really complicated, right? There are some studies where people have blown out, big areas of the epigenome using, CRISPR, and a clock won't change. So it's a very subtle turning on and off that I think is a very ordered and distinct way this happening that's much more subtle than we think. And so now we're looking at differential methylation analyses, differential transcriptomic analyses to do exactly like what you're saying. What changes and why? And now we can look at opposite ends of the spectrum, right? We can look at something that's aged or something that's younger versus older or has a different genetic or epigenetic background, and then try to tweeze out in our reductionist models exactly what's driving that. What's different that might be causal, and that's the game, right? That's the big game.
Fantastic. Hot on the trail. I love it. So what about using old, so in the culture media, right? That I get the argument about, well, you've derived them, these are young, these aren't old, but yeah, but they're from centenarians, right? But is it possible to take maybe plasma? I don't know if could you culture the neurons in the same plasma from the same individual as quote unquote an aged, environment?
Such a good idea. We should hang out more. These are all the right experiments, right? So we did this experiment where we made a series of mini brain organoids that we can make very standardized way from a couple of different lines. And these are mini sort of things that you can actually see with the eye in a dish. We can make an unlimited supply of those. And then we basically ran those in the normal culture media, which is a reductionist culture media, right? So it turns out to make an organoid, you have to back off. We were way more heavy-handed back in the day of making a specific red blood cell. You put in a crazy amount of cytokines, drive it to that. That's not how nature works. Now we let it be more unguided to make an organoid, for example, and that's a multi-tissue thing, right? So we have all these things. We have it in a really kind of basic culture media. These things can be grown for years, all right? And then what we did is we bathed these organoids exactly like you're saying with serum from centenarians that were the elite centenarians, people with ADRD, so Alzheimer's disease, or normal young and old serum. And remarkably, we're like, how long should you, this is the question, how long do you expose them for? How much do you put in? We made it up like we do most things in science, but we saw these tremendous changes. We saw changes in the necrotic activity of the cells. We saw changes in the epigenetic register. We saw some functional changes of improvement or a transcriptional signature where if you cultured a normal organoid with ADRD serum, suddenly that shifted to the transcriptional profile of ADRD. And subsequently, the opposite. If you cultured it in some kind of goodness that comes from a centenarian, you saw productive effects there. So we call that in vitro parabiosis, right? So the parabiosis experiments, I don't know if you've done a show on that. Crazy experiments. You link two mice together, the blood systems of two mice. Definitely efficacious, but really variable. We're trying to do kind of an in vitro version of that, where just like you said, we can swap up old and young, centenarian and not, to actually see. And that was kind of a proof of principle experiment that says, for sure, you can actually do this just by exposure to whatever might be in the serum or plasma.
Nice. Wait, so is that paper in process or in?
That paper is a million years off. We're kind of working out the conditions of how to do that. So Paula Arlada's lab came out with a paper, I think was in Nature per usual, that kind of showed that they did similar things where they exposed iPSC-derived brain organoids for like 2 years to different things and showed some interesting functional effects or defects there. So we're trying to speed this whole process up. The other thing is, one of my students said one of the most brilliant things the other day, which was, we're trying to figure out how to make something older. We're trying to do the opposite of what everybody else is doing. To try to uncouple it, we have to make our young system older. And a lot of things you try simply don't work. You try it, you think you're damaging the cell, creating senescence, creating hallmarks of aging, no change in the epigenetic register, no change in biological aging clock, maybe no change in functionality. So my student was like, maybe it's just time. And if it's time, then we're in big trouble. So if it takes 3 to 5 years for us to develop a phenotype of this, that's not going to work, right? So we need to uncouple these things. And it seems as if the serum exposure thing, it worked over a very short period of time, just a couple of weeks, right? So I think that has a lot of promise, but that is heavily developmental now. And so now we're working out, do you even need an organoid? Can you do it on a neuron? Can you do it on a triculture system? Or how reductionist can we make it and simplify it where we could multimerize it or do it at sort of high octane efficiency and run lots of different conditions?
Fantastic. The Murphy Lab is going to have so much great stuff coming out in the next few years. All right. So wait, hopefully we can go through, let's see, maybe I'll share the images so that, so I can have it open on my end. And then, because I think it will be informative for people, because when you think about, all right, wait, maybe I should edit out me doing this and then I'll talk about it in a minute. Hang on. So I'll share the picture and then if you could share that and I'll have it open on my end. Well, I'll see it on my end too, but.
Yep.
Oh, wait. Yeah, can't see. Yeah, just if you share. Okay, yeah. All right. So for those who don't know, if we're only looking at total white blood cells, this is generally an informative measure of quote unquote immune cell health because unless you're way too high or way too low, and plus, white blood cells as a group are a heterogeneous population of cells. So neutrophils and monocytes increase during aging and lymphocytes decrease during aging. So if you're only looking at total white blood cells during aging and their counts, you're going to miss those age-related changes, and then it's probably too late. But then we can even further subdivide lymphocytes into CD4, CD8, NK, B cells, and then you could even dig deeper into monocytes, which is on this chart here. Okay, before going there. So the importance of digging deeper for those who don't know, for lymphocytes, for example, in looking at CD4 cells, rejuvenating CD4 cells in Alessio Lana's group, granted, it's still in the preprint stage, but I'd be shocked if this data didn't end up panning out once it hits a publication, was that rejuvenating CD4 cells, CD4 T cells, which are a type of lymphocyte, dramatically extended lifespan, even to a magnitude greater than calorie restriction or any intervention, including rapamycin. So then it becomes, okay, what do these immune cell subtypes, these further subdivisions of lymphocytes and other cells, other immune cells, how do they look in centenarians, which you've got up here as EL, extended or extreme longevity, when compared with younger, middle, or older age groups? So I think I've shared this with George because this is from one of George's papers. I think it would be informative for people to see how these immune cell subtypes change during aging, especially for people who are interested in not just measuring their total lymphocytes, but also, commercially available labs. We can measure CD4, CD8, NK, and B cells, but then they're going to be like, what do I do with that information? How does it look in centenarians? What's good? What's bad? So I was hoping to get your thoughts, and I know most of that answer, so it should make for a good discussion. But it would be good to get your thoughts on how each of these things changes during aging, and then how centenarians either resist or it's further worse for them. So let's start off with the CD4s, because I think there's a growing body of literature where having them youthful and or rejuvenated, as I mentioned, is a big part of longevity. So let's go through what happens to the CD4s. Like the pattern is similar in the females and males. So if you could just walk us through what happens to CD4 levels and then how it differs in centenarians.
Sure. So I remember when we published this was a lot, right? So this is basically looking at proportions, right? But if you look at the overall sort of schema of blood cell and immune cell development, right? So I'm a hematopoietic biologist, which includes blood and immune cells. It's like my favorite thing. I think all the answers, including why these people might be long-lived people, has to do with their immune system, right? So to sort of take it back a step, we saw that there was a decreased ratio, as you just explained, of lymphocytes, right? That's a category that's like your T cells and your B cells to myeloid cells, right? So we always know that part of the innate immune system, that happens with age. So a normal change that you're going to have just sitting here is that decreased ratio of lymphocytes to your myeloid cells, right? So this was an expected change, but then, okay, in contrast to maybe the interpretation of this as far as the different proportions, okay, we saw that centenarians had more B cells. Okay, your B cells, explaining the way that you did, they're cells that produce antibodies. We'll just look at that in a very simplified way. And fewer T helper cells, right? So more B cells, fewer T helper cells. Those are cells maybe that help coordinate other immune cells, sort of activate to a particular immune insult, compared to younger people. Okay, so that change was not just an age correlated change, but it was a change that was unique to centenarians, male or female, right? And that did not occur in older centenarians, right? So I think I'm going to get to something that's really kind of controversial in a second, but our overarching thing, and this gets back to what I was saying about some of these people having had the Spanish flu and also COVID, right? So we think these types of shifts, right? When you put them all together, if you look over a lifetime of a centenarian, somebody that lives 100 years or more, this has trained their immune system to be faster, to be more effective to dealing with insult or infection, right? So if somebody's 100 years old and they get COVID, their immune system has been trained to an elite status for more than 100 years. And if you have more B cells, maybe, just maybe, they would respond faster to infections than T cells. Okay, the other thing that I'll say, which is related to having a more rejuvenated T cell, right? So if you look at cell numbers in centenarians versus non-centenarians, you had one of the striking things that we saw was you had more CD14 positive macrophage, that's a blood immune cell, and you had more cytotoxic T cells, right? So that's a distinct shift that another Japanese group who profiled centenarians, they also saw, right? And what this brings me back to is how you could have maybe it's not about the proportions, but it's about the fitness of the cells themselves, kind of what you were alluding to, right? So if you had a way to maybe rejuvenate your immune system, to reconstitute maybe the initial diversity of blood and immune cell types you had, that would be better, right? So this is where I'll introduce something called CHIP, right? So for your listeners, CHIP stands for clonal hematopoiesis of indeterminate, I don't want to use the exact nomenclature, but people don't know sort of where it came from, right? So conceptually, the idea is as you age, the diversity of your blood cell types becomes smaller and smaller. And why that is your hematopoietic stem cell, which is your master blood stem cell, fewer and fewer of those remain functional. So what happens is your entire blood system, which is trillions and trillions of cells, end up getting produced by fewer and fewer hematopoietic stem cells. And that is dangerous. That's normally equated with smoldering issues like CHIP, but with leukemogenesis, blood cancer, or with cardiovascular disease. So the less diversity you have, the more of your cells that are created from fewer and fewer hematopoietic stem cells, the more that's equated with mortality, okay? So when we went to go look at centenarians, okay, this is a very long-winded explanation, we thought for sure centenarians would have way less of this clonality, that they would have more diversity of their hematopoietic stem cells, and that would explain everything because they don't go through this clonality and badness that results in a normal person. Turns out centenarians have more clonality, and you can trace back some individuals that their current entire blood system is created from one or two HSCs, which you'd think, me as a hematopoietic biologist, is super destructive. Can't be good because you're going to have an accumulation of mutations and whatever. But the hypothesis we have now, and I have no data to prove this, but we're working through it, is just like you can have detrimental mutations that lead to leukemogenesis, blood cell cancer, cardiovascular disease as you age and as you increase this clonality, that you can also have mutations. So the one thing that we didn't see was these detrimental oncogenesis driver mutations in the centenarians that led to the clonality. You had this massive clonality, but it was driven by other things that we think might be making a more fit progenitor cell, which can then go on to produce a more fit T cell, kind of like what you're saying, right? So they kind of naturally might produce or rejuvenate their cells because the origin of their cells are from these elite HSCs that have increased functionality.
Stemness factors, I guess you would look at stemness factors in the HSCs, whether it's, I don't know, TCF7 or, I can go down the list, RUNX1 or.
Yeah, so that's one of the things we defined. The two people I started the CREM with, we were in Richard Mulligan's lab at Harvard, and that's what we worked on. We worked on side population staining, or what are the elite markers of an HSC. So if you look at a mouse, for example, okay, here's the interesting thing. If you look at a young and an old mouse has way more hematopoietic stem cells that you would mark by all the things you mentioned and all the markers of the classical markers of a bona fide blood stem cell. But even though in that aged mouse, you have way more HSCs, far fewer of them can reconstitute a mouse, right? So that's the gold standard. And why I love blood is that you can run the gold standard assay of gold standard assays, which is you can irradiate a mouse, kill its bone marrow and its blood system, and then reconstitute that mouse, if you wanted, with a single functional HSC, which would then repopulate the blood system of the mouse, right? So the really interesting thing is that even though you have more HSCs, they become less functional over time. So card-carrying, marker-bearing HSCs, but they don't retain that same functional quality of the cells that are in the younger mouse. And so you can imagine in a centenarian, you have some type of mutations, which we don't know what they are yet, but they don't seem to be pathogenic, that create a better, more fit HSC, which is kind of like maybe a younger or more functional HSC, which then goes on to make higher order CD4 T cells maybe, or cells that have more rejuvenative or the capacity to deal with or adapt to these insults, right?
So those HSCs, those one or two that are, somehow surviving in the centenarians, I mean, is that the golden ticket where if you figure out the mechanism for why they're maybe younger people, all of their HSCs are gone, they can't make the immune cells that they need, they can't be resistant to infection as a part of that story, and then they die, right? So is that how? Yep.
Yeah, and so I think that also connects into that concept of everybody loves the word inflammaging, right? So as you get older, remember we talked about that level of entropy or background noise, which kind of everything we look at in a centenarian is lower. We see that in the immune system as well, which is the more inflammation that you actually have, the more non-productive inflammation that you actually have, right? That's like basal inflammation that exists over a period of time. That's definitely equated with mortality, autoimmunity, cardiovascular disease, all these other things, right? So in a centenarian, if you have this much more elite system, which is more like a rejuvenated or young system, you're less perturbable with things that would create this kind of non-specific inflammatory response, which you don't want, right? So maybe that's part of the equation as well, is that you have less inflammation, you have a more elite immune system that has better CD4 cells that are not misprimed to sort of go off and, kind of inappropriately activate other sort of immune qualities that are off when they need to be off, but can regulate and can functionalize to a much higher order than you or I because they have this, rejuvenated quality of functionality.
Yeah. So wait, are you studying those HSCs from centenarians and to see what makes them functionally, I guess, elite compared to younger age groups or even centenarians who died at a younger age?
we're trying in different ways, and the way that we're doing it is kind of like a back end way. So we can create these sort of cell trees where it's a really cool process where with single cell data now, you can look at the repertoire of blood cells from anyone, from you or I, or from a centenarian, and that's a non-invasive thing. The way that we figured out how to make iPS cells was great because you didn't need to have a skin punch biopsy. You don't want a skin punch biopsy a centenarian, by the way, right? So we just take a very non-invasive blood sample. You can make an iPS cell, but you can also profile those cells and look in various ways to know what the origin of those cells was, right? Based on a bunch of different parameters I'm not going to get into, but you could trace back in a cell tree lineage sort of tracing way without, oligo tags or something to kind of understand the production of those cells, right? You can do uncoupling bioinformatically or something, right? So it's impossible, even from a normal person. I'm not going to capture your HSCs, which would be an incredibly invasive procedure. I'd have to mobilize you, and all those things are dangerous. You'd never do that for a normal experiment. And so obviously for a really sensitive centenarian, we can't kind of go in there. What we can impute sort of the clonality based on the outcome of the cells. And that is what we're studying. And so we make blood, right? So we can, going back to our origins, we're now trying to actually look at centenarians, recapitulating blood and immune cell development, and see if we see the same outgrowth of elites or differentiated cells where you have a different repertoire of cells. And that's how we'll study it. So we'll study it in the same sort of ex vivo model where we model blood cell development in a dish, which we hope recapitulates the way it's happening in the centenarians.
Nice. Awesome. All right, hang on. If I heard you correctly, I think you said there was an increased proportion of B cells in centenarians, but I think the B
Yeah,
cells here are purple, right? So if you look at
it's all the
young,
purple. Yeah.
yeah,
Yeah. So
yeah.
yeah.
So if you look at the total purple, it's 4.3%, just looking at the female data, 4.3, 7.1, 14.1. But then in the centenarians, it's only 3.5% B cells. So they would have lower B cells, but I think I heard you say they had higher proportion of B cells.
Yes. And so I think and that's why this is a little bit confusing based on the estimated proportions. But if you look at everything in aggregate, again, centenarians, and this is a centenarian specific effect, had like overall looking at aggregate, more B cells and fewer T helper cells, right? And so that change was unique to centenarians and wasn't one of the age specific effects that we use because we harmonized data over decades of every age group, right? So again, there's the normal changes that you would see, which are recapitulated in many systems of age. But one of the specific differences that we saw, more cytotoxic T cells, more CD14 positive macrophage and monocytes, more B cells, fewer T helper cells, right? So the conceptual thing there is maybe you had a more elite methodology of responding to insult or infection.
Yeah, I get that. Wait, and all of that makes sense on the plot because I see the M14 monocytes in pink, 9.1, 8.8, and then 11.1, 17.1, and the data increases also for the males too, right? So I get what you're saying about an increased proportion of the M14 monocytes, which makes sense. You're chasing maybe more microbes, systemically because the immune system is just weaker or whatever. But the B cells, I'm still hung up on the B cells because to me it looks like there are less B cells in the centenarians compared with at least the older age group, and even maybe at the same proportion as in young subjects. So, how
Yeah.
am I? Yep.
So I think what this is this was one of the things that we made in the revision. And so one of the things that we use the spectral flow for, which again is antibody mediated flow cytometry, in which you designate some of these individualized populations. And I know that because this was the only way that we could convincingly break down the different populations of CD4 and CD8 cells. So this may be in contrast to what we saw transcriptionally, right? So that could be one of the reasons why it was different. But again, proportionality versus the overall number of things that we saw in aggregate across things, maybe transcriptionally, might be the reason why it looks a little different.
But the reason I hear you there, but if it's so if it's really that B cells increase during aging, which would be the opposite of what I see on the plot, the reason I'm raising that issue of B cells is because there was a paper that was recently published that showed that B cells drive CD4 immunosenescence, and then that's potentially important because, as I mentioned earlier, rejuvenated CD4s are a part of that pro-longevity longer than every other intervention in mice. And also calorie restriction dampens down B cell levels, which suggests that maybe if in EL they have lower B cell levels, they'd have less CD4 immunosenescence, and maybe that's a part of their pro-longevity approach. So am I misinterpreting that, or what are your thoughts?
No, I mean, I definitely hear that, and I'm definitely on the same page as far as the concept of a rejuvenated CD4 cell, right? And so, in that complicated explanation I gave about clonal hematopoiesis, which is normally seen as a bad thing, I think maybe in centenarians there's evidence that suggests that it could be causal in a productive way. And one of the ways that would result is that you would have more functionalized. Rejuvenated is a word that everybody uses. It's a hard word when you're looking sort of at steady state in this conceptual idea, but maybe the cells that you're creating from these elite HSC starter cells are resistant to aging, or basically have a more rejuvenated quality, or come from a higher order HSC that's more functional to begin with. And you had kind of alluded to that, right? So you end with the same result, which is a more elite CD4 cell that doesn't have sort of the age associated properties that would lead to, deterioration or something. So
So
you
that would be
you have kind of in-house rejuvenate an in-house rejuvenation or de-aging program based on the genetics of your HSCs that are producing the daughter cells downstream.
so that would be a pathway independent of B cells negatively impacting CD4s.
Yes, but would still end up with the same kind of result, where if you're, eliminating some of the non-functional CD4 cells, basically through a kind of a targeted senescence pathway that leads to some kind of rejuvenation, it's a similar sort of thinking as far as how you would end up with a more elite immune system.
Yeah. So
But yeah,
then, so
I,
then Nice. So cytotoxic T cells was a new one for me because you can see, so the cytotoxic T cells would be in dark blue. And if you look at it at younger ages, at all younger ages, I mean, it's virtually absent at young, 0.3%, 1.5%, 2.6% in the females, and then it jumps to 7.5%. So I mean, that's a new concept to me. So it's got to be a new concept to most people. So what's the specific role of cytotoxic T cells that there's this dramatic expansion in the centenarians?
yeah. So again, I think it's a super interesting and maybe one of the things we're thinking about with centenarians, and what's really interesting for me as a non-geneticist, is there doesn't seem to be a magic bullet, like a one particular thing that leads you on this pathway to being a centenarian. So we see a good amount of heterogeneity in the population. So now we're thinking about this multiple paths to sort of maybe getting to elite immunity. So I think one of the ways you could also hypothesize that you could also get this elite immunity is that, in the basal state, you have very less basal activity, no smoldering, inflammaging that leads to something. But then maybe you have, these cytotoxic T cell soldiers that are these elite soldiers that you have more of, and you have more diversity and more sort of a repertoire of this particular population in a centenarian that doesn't decrease the way it does with other people. And these things have this additive elite way to sort of deal with some of the insults that you have over time, right? And you just retain the quality of these cells to respond to these emergent situations like COVID or the Spanish flu or things of that nature, right? So I think the challenge is understanding exactly how you retain this elite immunity. And I think for us, the conceptual idea is that it's not just a genetic thing and you're bestowed with this amazing immune system that just works this way, but it's adapted itself. It has sort of the underlying genetics and propensity to be able to train itself over decades to respond in a very different way, like this elite way in which it's very specific. It doesn't respond until it needs to, but when it needs to respond, it responds with, efficacy and power, because of the fact that you have the repertoire of cells that you need, and you are different for some reason. You have more functional properties, maybe. It's an
Yeah.
interesting.
Oh. Yeah. So definitely along those lines. And it may be a simplistic experiment or a simplistic thought, but in terms of those CD4 cytotoxic cells that increase, and, with the mechanism you just mentioned, that they're maybe a part of that phenotype that really helps give them that ability to chase down infections and respond to insult. Did you look at associations between the levels of CD4 cytotoxic cells with the things that increase during aging, like, for example, the NK cells, which are in green? You can see that they jump to 20.8 and 28.9% in centenarians. And then also, the M14 monocytes, which both of those cell types would be predicted to be, quote unquote, pro-inflammatory as they're chasing down microbes. So I wonder if the ability to have higher levels of CD4 cytotoxic cells could be a compensatory mechanism if your phenotype is JFC, I'm like talking in text speak, JFC, like we've got this massive burst of NKs and monocytes. We're chasing down microbes like crazy. But if we have this burst of the CD4 cytotoxics, maybe that's a part of counteracting that. Whereas centenarians that might not have as big of a robust CD4 cytotoxic response, maybe they die off sooner. So have you looked at maybe associations between the cytotoxic and the levels of the NK and the M14 monocytes?
We have not, but I love your thinking, and I think the immune system is so complex. It's elegant in its complexity and simplicity, really, in some instances when it responds, right? So again, one of the new papers that we have coming out, that actually is coming out and has been in review for what seems like 100 years, is a paper in which we could actually make not just T cells, but we can make both CD4 and CD8 T cells. And we're also trying to make very specific subsets of T cells that would sort of phenocopy the functional properties of the cells in you and I, right? And so we think we're all the way there. It was a slog to sort of go through these different developmental ways. It turns out it's really easy to make an NK cell, and everything that you actually do from an iPS cell just trips to NK identity. You have to kind of steer the ship in a completely different direction, not just to get a CD4 cell, but to have a cell that's capable of then going through switching and sort of repurposing and reprogramming itself at that point. So now we think we can do that. So again, one of the things that we think the system might be good to do is not just to model the dynamics of maybe how HSC growth and development might be different in centenarians, but now we can actually make B cells, T cells, different subsets of CD4 and CD8 cells to try to understand maybe how this is all working. Like, can we create an ex vivo model that could then be imported into a mouse, and to look at sort of reductionist ways of how the functional properties of these cells might be functioning in the people. And again, it's a very reductionist way, but now we can make many of the different subsets that might be involved here. We know how to make macrophages, for example. So we can now make, after 10 years, maybe the entire repertoire of the immune cells that we might need to model exactly maybe what we're seeing here. And then, of course, we have the iPS cells from these elite responders to see if we can recapitulate some of these dynamics, maybe outgrowth of those, cytotoxic T cells versus some of these different proportionality. Does that happen if you let it play out in a dish?
Nice. So just to reiterate this for people who don't know. So recent paper published, proteomic-based clocks for all of the major organs: kidney, liver, intestine, muscle, brain, immune. And of those organ systems, the two, and I get that most people think muscle is the organ of longevity, and I get that. I'm with that. For people who don't know, George is fit too. I've seen you on your channel. You take care of yourself physically. So physical fitness is a given for people in the longevity space. But then how do you dig deeper? What's under the hood? So your lab is uniquely suited. Like, I can't say this strongly enough. When you consider that the brain and the immune system, in addition to muscle, of course, it's important, but brain and immune system were the two top hits for predicting a longer health span and longevity. So it's like, I'm super excited to see, all the great stuff that comes out of your lab, because you're literally doing all of that: brain, immune, and in centenarians, and at the single cell level, and testing these things molecularly. So it's good stuff. All right, so we have another question. It's more practical, and it's, less esoteric, and I think most people will enjoy. And, so given you're swimming in this pool of longevity and studying aging, like, are there, besides the standard diet, exercise, sleep, are you using any of the unique insight that you've developed to try to optimize, actually to track and potentially optimize your own, whether it's your own immune cell subtypes, immune cell function, HSC levels? I don't know if you're tracking these things on your own in the lab. I'd probably be doing that. But are there any tricks of the trade that you're using in addition to the standard eat real food and exercise?
Yeah, and so and you can talk to my people. So obviously, I'm an N of one, and so obviously there are the Brian Johnsons out there. So from a Brian Johnson point of view, and I think everybody knows who Brian Johnson is. He's kind of like a tech guy who now has like a $2 million lab in his house. He kind of goes through the thing where data is what's going to drive everything, but he also kind of goes off the rails a little bit there from a non-scientific angle, right? And so that's what we try to do. Part of the sort of role of the creme is to do very rigorous science. And so with that said, me as an N of one, of course, it's really hard not to try to sort of create this data sort of for yourself. So obviously, I am plugged into all of the clocks that we actually run. I have longitudinal blood samples that I keep sort of metrics for. Pretty much every assay that I've ever sort of run or optimized in the lab. Even now at full professor, I don't know if my people enjoy this as much, but I definitely still run some of my own experiments. When I'm back in the lab, everyone's like, oh man, maybe he knows how to do this, maybe he doesn't. But I think it's important to sort of stay connected to that. So a lot of the experiments that I actually run, I'll run on my own cells. Like blood is amazing, whereas if you needed platelets, you could take a drop of your blood and there's millions of platelets there to run sort of signaling assays to look at aggregation or things of that nature, right? So for sure, um, conceptually, even on my own iPS-derived cells, I am, of course, looking at maybe how my cells might respond to geroprotector A or sort of geroprotector B, right? So trying to understand by design if conceptually this thinking of what gets recapitulated in the dish or how it might respond there might respond in an N of one of me, right? And so one of the things that we're looking at now, as far as what are the geroprotectors, are things that are nutraceuticals. So like even Rich Miller and testing in mice, people are looking at things like astaxanthin. Astaxanthin shows an incredible, at least in male mice, an incredible increase in longevity in mice, right? So obviously that's something you can order from Amazon. So things that have a lot of scientific background, and you and I are the same. We love data. I always say knowledge is power. So things that I feel have a strong scientific background that have been replicated across sort of multiple laboratories. Lots of times people would say these things haven't been tested in humans, but what really has? Not much. And so things that I think are safe, that are sort of readily accessible, that are not sort of drugs that you need a prescription for, I take a bunch of those supplements. And those are things that I'm coordinately testing in my own cells in a dish at the same time.
Wait, so can you share? Are you happy to share what you, yeah, because people
Yeah,
want to
yeah.
know, yeah, the experts, what are the experts in the field doing, So,
Sure. So, like, I think, like many people in the field, NAD is a complex one, right? And depending on how you talk to, I think the jury is still out. There's a lot of data more recently that if you look at very specific effects, NAD is known to massively decrease with age. So, depending on which field you're in, if you're a David Sinclair or a Brenner person, are you taking nicotinamide riboside or NMN, things of that nature? I'm not going to say which one I take, but I take one of those. Okay. So I think that that might be important for you and I. What's really cool is the end result of tryptophan metabolism is NAD. So there, it's really interesting that that's kind of cooked up in there as well. So I take an NAD precursor. I take, I'm a big alpha ketoglutarate fan. Okay. So I'm a fitness person. I'm a weightlifter. So a lot of things that are connected to that alpha ketoglutarate. I should say that in the most recent ITP that showed no efficacy, right? So it doesn't seem to do anything in mice. So, I see that as kind of like a null-sum game. Things like vitamin D. So I have the ability as a hematologist to run my blood through like complete blood counts or just looking at dynamics of different things. So I try to sort of keep track of those things. One of the things that I noted, and I now have all my friends do, is look at their vitamin D level. A lot of people, particularly, it's been a really rough winter here in the Northeast. I should move to Austin. But it was terrible. I'm from California originally, so obviously that takes a long time to be depleted, but my vitamin D is very low. So I basically do vitamin D supplementation. And so I'm always on the lookout for things of that nature. Mostly it's these things that are relatively benign, but certainly I don't know if I'd ever be on a GLP-1 drug, but we're looking at, for people that don't know, we can order a GLP agonist mimetic from a company and just get a small amount and test that. And so I really think the results we're seeing, where we're putting GLP-1 agonists into neuronal cultures, is significant. I'll be showing you that data at some point in some of our collaborative meetings where it has specific effects. So people have not known, obviously these are miracle drugs, but as a potential mediator for the prevention of cognitive decline, I think there's a lot of potential. The jury's still out there. I don't currently take a GLP-1 drug, but maybe at some point everybody should. So we're on the lookout where we're trying to not only model these things, but also understand what is a geroprotector. I still don't know if we know what one is, right? And so if you don't have an underlying disease process, but you take a GLP-1 drug, is that a geroprotector then, right? If you take something that fixes an underlying pathology that you have, is that geroprotection? Is that a longevity signature, right? So I think I'm an experimental person, and as I go through these things, as we continue to define what is what, I could see myself, adapting and modifying, optimizing myself in different ways depending on what the data shows for sure. I think that's part of the fun of it, right?
So the big ones for you are NMN or NR, vitamin D, alpha ketoglutarate, even though it showed no effects on lifespan. Are there any other supplements or things you include in your diet that may be quote unquote immune enhancing or lead to a specific immune cell subtype profile that maybe you're looking to keep the immune system, healthy and fresh?
Yeah. So during COVID, everything was shut down, and I came up with this. Now look, it seems like a crazy plan at about 3 o'clock in the morning, where I texted my grad student at the time and said, "Hey, the government's having problems with COVID tests. We should just build one in the creme, like MacGyver." So we came in and we did that, and we had a CLIA certified test, the first one that was around, that we built in the lab from just RNA extraction kits and a bunch of stuff that we knew how to do in the lab with our own oligos, everything. So then we started running all of the COVID tests for the hospital here. And at night, my people would get on the internet and describe to other people the blueprint of how to do this, because the COVID test was a disaster. Nobody, our friends and colleagues, nobody could be tested. During that time, my trainees, we were the only ones around in the lab, right? Nobody had any masks because there was no masks. I would ask them what they want to eat, and it was always chicken McNuggets and Coke Zero. So I ate chicken McNuggets and Coke Zero. At the end of that tour, when we finally got out of the testing business and things kind of went back to semi-normalcy, I was like, you know what? Not only am I never eating another chicken McNugget, I'm going to be vegetarian. And so I'd always kind of played with that idea. So literally for the last 5 years or so, a little bit more now, I've been vegetarian. Did I do that because I think that's going to make me live longer? No, but it just seemed like a logical adaptation. I'm a very sort of disciplined and structured person. That's how I've always been. So a lot of this stuff really fits into my scheme of how I think about life in general, and how every day I kind of do the same thing, right? I get on elliptical where I go through my cardio, that's fasted cardio, before I do X, Y, or Z. So I lead a more increasingly structured life, which includes my supplementation, how I do everything else. And being a vegetarian just kind of fit into my lifestyle. But definitely, there's a correlation between processed foods, red meat, and some of these other things that probably being a vegetarian is not a terrible idea.
But looking at specifically the effect of whether it's supplements or a vegetarian diet on immune cell subtypes, even things like PD-1 expressing T cells, T cell exhaustion, or autophagy. You could look at, I think it's LARG3. You can look at autophagy in immune cells. So not doing that yet, because that's what I'd be doing. I don't know. Not
Yeah,
yet.
not yet, but definitely it's a lot of interest in testing these different parameters of things. Right now, basically, I'm putting my cells through all of the different functional assays. So I've run the mitochondrial assays, I've run the cell response assays, I've run biological aging tests, I've run different dynamics of functional properties, directed differentiation. And so it's more of this non-granular way of just looking at how we can sort of interpret and look at the functional differences between cells. And it's interesting to see how my cells perform in comparison to other things. And there's definitely unique things there, right? Where, what is a control and what is wild type and what is a normal response versus an abnormal response. I definitely see, we've been running these MSD tests, like mesoscale tests, where you can take a tiny bit of serum. In our case, you can take a tiny bit of cell culture supernatant and run it through a cascade, a very nice panel of antibody mediated inflammatory markers, right? And definitely the way that when I LPS stimulated my cells, I see a really differential response when I look to other controls, right? And I don't know how to explain that, but there's a very interesting response that I see dynamically as far as some of the chemokines that actually come up than other things. And so that just kind of connects us back to the home, which is we've been selling as the creme and as an IPS outfit, that the way to do all of this is from a personalized approach, that everyone is an individual. And the more that you can individualize, not just disease therapies, but maybe some of these rejuvenative or geroprotective agents, has to be done on a personalized basis, right? There's that big paper, which is very controversial in Nature, that talks about how you might respond to one of these GLP drugs has to do with your genetics. So there's a lot of controversy about it. But I think the underlying message is probably true, that all of these things, the more that you can look at it from an individualized point of view, the better, right? So I continue to try to not understand the biology of aging, but understand the biology of aging that's happening within myself.
Fantastic. All right. That was great, George. I'm looking forward to hosting your work on the channel again at some point. And I think I'm going to be on your channel too. So yeah, looking forward to that. And yeah, have a great day.
Mike, that was awesome. I appreciate you. Have a great one.
Bye.
Bye.